Good afternoon, ladies and gentlemen. This is microbiology lecture. Causative agent of tetanus and then causative agent of anthrax. And it is the last lecture, the last microbiology lecture this term. Causative agent of tetanus. Causative agent of tetanus. The first one, causative agent of tetanus. Latin name of the causative agent of tetanus is the Clostridium tetani. Tetanus is an acute wound, toxinemic infection accompanied by clonic and tonic fissures caused by the specific Clostridium tetani, genus Clostridium, family Clostridiaceae. Morphology. Vegetative cells are straight rods with rounded ends, mobile, peritrichous, and have terminal spore, drumstick rod. In speed, they are ended randomly or in chains. According to the ground, they are positive, gram-positive rod. There is a picture of them. Cultivation. Clostridium tetani, like all Clostridias, are obligate anaerobes. So they are not able to multiply in presence of O2. And on nutrient agar, they form R-type colonies or sometimes S-type. In a column of agar, fluff or lenticular growth is formed. Of course, RCM broth is used for cultivation of Clostridia. Robertson cooked meat broth is used as usual, and they develop uniform turbidity. Biochemical properties. Biochemical activity is low. Weak proteolytic properties slowly break down proteins and peptones to amino acids. Antigens. O antigen, H antigens, and for 8 antigens, 10 serovariants are isolated, not important in practice. Of course, exotoxin is recognized like protein antigen, and specific antibodies are produced for exotoxin, antitoxins, and very important in case of toxinemic infection. Virulence factors. Virulence factors, of course, is exotoxin. This exotoxin composed of two substances, tetanus plasmin, it is neurotoxin, and tetanolysin, it is hemolysin, tetanohemolysin, cause destruction of the red blood cells. Tetanus plasmin is neurotoxin, and it acts on nervous system, on synapses. So they remain exotoxins, and they are produced in human body, and they are circulated in the blood, and circulation in blood of exotoxins is named toxinemia. So tetanus is a kind of toxinemic infection. Pay attention, the exotoxin is the keyword for all disease caused by Clostridia. All Clostridia cause toxinemic infections, including tetanus, botulism, and gas gangrene. Resistance. Spores are resistant to low temperatures, heating, and spores are inactivated just by autoclaving at temperature more than 130 degrees for 20 minutes. Vegetative cells, of course, die on boiling for 5 minutes, but very difficult to destroy spores. Spores of all Clostridia are spores-forming rods, spores-forming bacteria, and it is very difficult to destroy spores. Epidemiology. Really Clostridia, all bacteria of Clostridium genus, they exist in the intestinal tract of human beings, animals, and birds and fishes, like rods, like gram-positive rods. And they go out by feces constantly, and in the soil, they are able to multiply a little bit, but there are no favorable conditions for them. And finally, all Clostridia develop spores. So spores formation is very important information about Clostridia. And the source of infection is soil because of spores of Clostridia, and all people belong to this group because we are in contact with soil. Particles of dust are enough to enter spores. So the infectious process is started from the entering of spores. And transmission mechanism may be wound infection. Tetanus very often is wound infection. Contamination of the wound with soil or through contaminated dressings, medical instruments. In this case, we are talking about iatrogenic infections, when occasionally the patient was infected by bandage, for example. Bandage should be sterilized, of course, but sometimes maybe the quality of sterilization was not good. The temperature was not enough to destroy of spores of Clostridia, and really, but sometimes due mistakes of doctors, iatrogenic infections are developed. It is possible to develop tetanus in newborns with infection of umbilical cord after delivery. Sometimes, if the mother did deliver a baby in aseptic conditions without any medical aid, except the maternity hospitals, newborns tetanus is very often in developing countries especially. The clinical manifestations. The incubation period lasts from 1 day to 2 weeks, and the disease begins actually. This is acute diseases. The first symptoms are the spasm of chewing muscles, and the sardonic smile is developed. Patient is not able to stop this smiling because of spasm of muscles. Firstly, muscles on the face are contracted, and then other muscles slowly contracted. The disease progresses, and muscle spasm of trunk and limbs, epistotonus. Well, this is epistotonus of newborn, and this is very famous picture of some British soldier. He did die in 19th centuries during some war, and finally the body is killed, like because of spasms. And diagnostics, microbiological diagnostics, material for research, contents of this is we should take some liquid from the wound, pus, maybe pieces of tissue, maybe exudate, bandage if the bandage is source of infection, then blood. For neonatal tetanus, material from umbilical channel. Bacteria are usually found at the site of entry into the patient's body. First method that is used, it is microscopical method. The smear is prepared from the test material, stained by gram method, and we are looking for gram-positive rods, thinly arranged. Cultivation. Cultivation is used, and isolation of this bacteria is carried out by inoculation on Robertson cooked meat medium. It is a broth, and you know we did study this medium. It is a broth, nutrient broth plus glucose and plus particles of cutted meat, or maybe some internal organs of animals, and the meat is used for oxygen absorption. The medium should be heated, boiled really before inoculation of the specimen to remove O2 from this medium. And then we have to wait till the human body temperature, and then the specimen should be inoculated on this medium. And then we should re-inoculate bacteria on blood agar in Petri dishes. And these Petri dishes with blood agar are incubated at thermostat in anaerobic conditions. Now you should remind methods of cultivation of anaerobic bacteria, like maybe Macintosh and Phil's jar, maybe gas pack should be used, maybe exsicator. So for cultivation of bacteria if they grow up on Petri dishes on blood agar. And then pure culture should be isolated again on Robertson cooked meat broth, and it is identified by morphological, staining properties, cultural, biochemical properties, and detection of toxins. Toxigenicity is also determined. For express diagnostics, immunofluorescence test is used. Biological message is used for detection of exotoxin, because the clinical symptoms of tetanus are developed due to exotoxin action. So the first task is to detect exotoxin. To detect exotoxin, and mice are used, and special test that is named neutralization test is used. In this test, we are talking about neutralization of toxin by using of antitoxic sera. Fortunately, all strains of Clostridium tetani produce exotoxins with the similar antigenic structure. They produce similar toxins. It is easy. We have no difficulties in diagnostics, and we have no difficulties in vaccine production. So one antitoxic sera is used in diagnosis of tetanus, and it is used for neutralization test. We should prepare a mixture. So what is the aim of neutralization test? The specimen, we should use 2 groups of animals, control and experimental, and the specimen should be injected into 1 group of mice, just specimen where probably exotoxin is, and then we should prepare a mixture of the specimen and antitoxic serum where antibodies are for tetanus exotoxin. And this mixture should be injected into another group of mice, and we will exactly into the leg. And after injection, if we will see after injection of the specimen from the patient, injection of this specimen into the leg of mice, if we will see the spasm of muscle of the leg and so contracted leg, and then we will see strict tail, there are clinical symptoms of tetanus. And if such symptoms are not detected in the mice that did get injection of mixture of material and antitoxic sera, we can make conclusion that exotoxin is detected, and the treatment should be done as soon as possible to save the life of patient. Cultivation method is used if you are going to check the quality of sterilization of bandage, for example. In this case, we are looking for spores. So it is possible to detect spores just due to cultivation of the specimen. A serological method is used for detection of toxin. ELISA test is used for detection of toxin now, and coagglutinating test is used. So the first aim is to detect exotoxin. Treatment. In case of toxinemic infections, when the person is going to die due to exotoxin action, the first aim is to neutralize exotoxin. So for neutralization, we realize that chemicals and antibiotics are not able to neutralize exotoxin. And the only medicine that is able to neutralize exotoxin is antitoxic serum. So it is possible to use, unfortunately, horse's serum is used, anti-tetanus serum is used for treatment. Of course, it is better to use human immunoglobulin, homological. If donors are available, if they are immunized by anatoxin, it is possible to get finally purified immunoglobulin from hyperimmune serum or blood of human beings. But we have no donors enough. Sometimes they're not agreed to be immunized. So horses are available, and horses are immunized by an tetanus anatoxin, and because this serum should be produced. Tetanus is very common infection, and all medicines have expiration date. So when after expiration date, even this serum was not used, it should be destroyed. And so this antitoxic hyperimmune serum is produced constantly every month. There are special microbiological institutes, and they should produce this human horses anti-tetanus serum if donors are available. the purified preparation immunoglobulin is produced, okay? At the same time, antibacterial medicines are used, and the Clostridia is sensitive to chemicals such as metronidazole and tinidazole. Really, these chemicals, you have to know, they are used for treatment of disease caused by protozoa very often. And it was detected that strict anaerobic bacteria are sensitive to the same preparation, so they are used. But the first line preparation, the first medicine that should be used, it is antitoxic serum or immunoglobulin. These medicines content of antibodies for exotoxin, and they are able to neutralize exotoxin. A specific prophylaxis. So in case of toxinemic infections, of course, the purpose is, the aim is to produce antitoxic immunity. So for production of antitoxic immunity, microbes are not used. So no killed vaccine, no live vaccines, because we should create antitoxic immunity. And for antitoxic immunity, toxoids are used. And toxoids are used for specific prevention of 7 toxinemic infections. And for pharmaceutical faculty students, very important to know medicines, what medicines are used. So in case of 7 toxinemic infections, toxoids are used for specific prophylaxis, like staphylococcal infections, then disease caused by Clostridia, tetanus, botulism, gas gangrene, then diphtheria, then whooping cough, and cholera. So for specific prophylaxis of these 7 infectious diseases, toxoids are used. For specific prophylaxis of tetanus, toxoid is used. Toxoid in English, anatoxin in Latin. So, and it is produced like monopreparation, but really specific prophylaxis of tetanus is done all over the world by vaccination schedule. And this tetanus toxoid is included in triple vaccine. This triple vaccine is named DPT, and it is associated vaccine. Triple means this medicine consists of 3 different vaccines, and they are injected together at the same time for production of immunity against diphtheria. D means diphtheria, P means whooping cough, and T means tetanus. So it's not one vaccine. This medicine consists of 3 vaccines. They are joined. And nowadays, DPT, modern DPT, content of 3 toxoids: diphtheria toxoid, whooping cough toxoid, and tetanus toxoid. In past, DPT was used, and the prophylaxis of whooping cough was done by killed bacteria. And this vaccine is used at some countries. And now 2 kinds of DPT are used. DPT new old vaccine, when for specific prevention of whooping cough, killed bacteria are used, and new one vaccine content of 3 toxoids: diphtheria toxoid, whooping cough toxoid, and tetanus toxoid. And maybe DPT just with low concentration of antigens. It is produced in Russia just for some children with some disorders, neurological disorders, for example. And non-specific prevention. Non-specific prevention is at first medical aid. The surgeon should examine the patient, and if the wound is big and there are necrotic tissues, surgeon should remove necrotic tissues to avoid anaerobic conditions for Clostridia multiplication. So this first medical aid is done for prophylaxis, non-specific prophylaxis of all diseases caused by Clostridia. So I'm repeating, for specific prophylaxis of tetanus is done during the first year of life. DPT is injected in Russia. The first vaccination is done at the age of 3 months of newborns. Then a few revaccinations till the age of 18 months, the children will get DPT. And then after 18 months, whooping cough is not so dangerous. But people are infected by Bordetella pertussis, but they don't die. But diphtheria and tetanus still dangerous for adults. And in Russia, by Russian vaccination schedule, vaccination is continued against diphtheria and tetanus by using of 2 toxoids DT preparation without P. DT preparation is used like here. This is anatoxin, consists of diphtheria toxoid and tetanus toxoid. And an adult should be vaccinated by Russian vaccination schedule every 10 years. At the age of 16 years, 26, 36, 46, because tetanus is and diphtheria still dangerous, okay? And then, so absolutely healthy people should be vaccinated. If we are talking about specific prophylaxis, we are talking about prevention measures. Absolutely healthy people should be vaccinated for any case. Maybe they will be infected, maybe not. And then we are talking about emergency prevention. If some person has wound and he will visit a doctor, doctor should suggest to do emergency prevention of tetanus, emergency immunization. So in this case, we are talking about combined immunization. The tetanus toxoid should be injected at one part of the body, maybe on right arm, for example. It is named active immunization, but it takes time for immune system to develop antibodies. They will be developed in 1 week. So we are going to protect this person from tetanus exotoxin by using of antitoxic serum. It should be injected at another part of the body. This procedure is named passive immunization. So active immunization plus passive immunization, the procedure is named combined immunization. It is done in case of emergency prevention for people with wound, if person has some wound. That's all. And now we will continue, and I will give you the lecture, causative agent of anthrax. You will get 2 lectures at one day. So the causative agent of anthrax is named Bacillus anthracis. By morphology, they are gram-positive rods located in smear in pairs or maybe chains. Do you see they are located in chains? So they are named bamboo stick by this location. Immobile, they have no flagella, form spores centrally located. I hope you know that bacillus and Clostridia are spores-forming rods. And Clostridia develop bulged spores. Bacillus develops endospores. Those are located inside the cell. Cultivation, aerobes. So they are cultivated in presence of O2. And they grow on simple nutrient media at temperature 35-37 degrees by Celsius. And they develop R-type colonies, looking like head jellyfish or lion's mane, they are named. Or they're a picture of Bacillus anthracis colonies. Really, they don't need blood agar. There is photo of colonies on blood agar. Blood agar is used for identification of these bacteria because they have no hemolysins. And after pure culture installation, they are inoculated on blood agar, and we have to see that they don't develop hemolysins. They do not destroy red blood cells. On liquid media in the broth, sediment appears at the bottom of test tubes, looking like some particles of cotton. Biochemical properties, biochemical activity is high. They are able to ferment many sugars, and proteolytic enzymes are produced also. Antigenic structure. There are 3 main antigen groups. 2 of them are capsular antigens and a toxin. Capsule antigen, somatic antigen, and toxin, protective antigen, lethal factor, and edematous factors. They are named like factors, but really they have toxic properties, all of them. And because of production of these factors, sometimes lethal outcome is developed very soon in case of anthrax. Resistance. Again, spores are highly resistant. They die at just 140 degrees by Celsius in 2-3 hours. And they remain in water for up to 10 years, in soil up to 50 years. 50 years, maybe 60 years, so they're very resistant in nature, spores. No, anthrax is not eliminated from the earth in spite of nowadays vaccines are developed, animals, farm animals are vaccinated, people, those take care of farm animals are vaccinated. In spite of, not possible to eliminate anthrax because of spores. They are able to survive in the soil for many, many decades, even for till 100 years. And time by time, sporadic cases of anthrax are developed. Okay, and epidemiology, how anthrax is developed. Anthrax is a typical zoonosis. It is zoonotic infections. And the causative agent is an environment in the form of spores. Animals, grass-eating animals, by eating of grass with some particles of dust with spores are infected. And bacteria germinated from spores, and bacteria multiply in the animal's body, like cows, for example. Very often cows are infected. And bacteria go out with all biological fluids from this animal, and again in the soil they are able to develop spores. So spores are able to survive in the soil again and again. So time by time, new spores are developed, are produced, and people, those take care of animals, belong to this group, of course, firstly, and then a person becomes infected by contact with infected material, who takes care of animals, maybe who works with the skin of these animals to do some goods from the skin of animals. And people, those are far from these farm animals, are infected by eating of the meat of sick animals if it's not properly cooked, or as well as by inhalation of spores, if they have to prepare some goods from the skin. So there are different transmission ways. Direct contact, then by food, eating of meat, and maybe inhalation of spores. And so clinical manifestations. The incubation period is from 2 till 6 days. And there are different clinical forms in cutaneous anthrax, pulmonary anthrax, and gastrointestinal anthrax. It depends on the route of transmission of causative agent. So cutaneous anthrax, if people are infected due to contact with animals, so cutaneous anthrax is developed. It's not very dangerous clinical form. The lethal outcome is not developed very quickly. So at the site of the entrance gate of infection, a red spot first appears, quickly transforms into a papule, and then a vesicule is formed, which is very itchy. And then we will see brown-black scab. So the disease is named anthrax because it is looking like charcoal, okay? And pulmonary anthrax. Pulmonary anthrax is develops with inhalation of spores, and it is extremely difficult. And with the development of intoxication, pneumonia, and pulmonary edema, death occurs in 2 or 3 days. Okay, very dangerous, very severe clinical form, pulmonary anthrax. And then gastrointestinal form. It is characterized. It is developed after eating of meat if it's not properly cooked. Like steak, middle red with blood, for example. Are you sure that this animal was not infected by anthrax? So some people prefer the steak with blood. Meat should be properly cooked, okay? So it is characterized by the development of general intoxication, vomiting with blood, and diarrhea with blood, And the death of patient occurs in 3-4 days from heart failure. Not possible to save the life of patient if they have gastrointestinal form of anthrax, very severe. And then microbiological diagnosis. The material for research depends on the form of diseases. So maybe we have to take some specimen from the carbuncle, from this ulcer. Maybe blood. Septic form is developed with bacteria multiply in the blood. In pulmonary anthrax, sputum should be exempt, of course. In gastrointestinal anthrax, feces, vomiting mass, and so on. All samples must be placed in sealed containers and transported in a sealed box, of course. It is a dangerous infection. Microscopy, conventional infection. Microscopy for the test materials smears are prepared and stained according to the Gram method. We are looking for gram-positive rods in chain. Cultivation is done, is inoculated on simple nutrient media and R-type colonies. We are looking for R-type colonies. They should be suspicious. And then the pure culture should be isolated and should be identified as usual by using of biochemical property, then absence of hemolysis zones, and bacteriophage is used for identification. Again, and some biochemical properties. Serological method is used for rapid diagnosis. So by gram staining method, not possible to identify bacteria. We are not sure that they belong to Bacillus anthracis species because of Bacillus genus includes many species. Of course, for rapid diagnosis, immunofluorescence test is used. Direct immunofluorescence test, IF test, is used for detection of bacterial specimen. And in this test, on the one hand, it is a microscopical method, but on another hand, of course, it is serological method. And in this case, the smear should be prepared from the specimen that is sent to the laboratory. The smear on the glass slide, the smear should be fixed, and after that, the serum, a diagnostic serum, fluorescent diagnostic serum, the drop of serum should be placed on the smear on the glass, and this glass slide should be, the smear should be incubated at thermostat for 15 minutes, because it takes time, and diagnostic fluorescent immune serum against anthrax, content of antibodies for Bacillus anthracis. So if in the smear there are Bacillus anthracis, antibodies will react with this bacteria and they will be fixed on the slide. After 15 minutes, the slide should be washed and we should remove excess of antibodies, and not possible to see result by eyes. Result is not visible. So to see result of immunofluorescence test, special microscope is used, and it is named fluorescent microscope. And a fluorescent microscope has a source of ultraviolet rays, the lamp of ultraviolet rays, and by immunoglobulins are conjugated with fluorochromes. And every fluorochrome has ability to get some light, the 13 lengths of wave, and to get this light and then to give lightning. And it is possible to notice this lightning by using of fluorescent microscope. And really, in case of anthrax, we will see rods in chain lightning. The color of lightning depends on the fluorochrome that was used. Very often fluorescein isothiocyanate is used like fluorochrome, and we will see green lightning. If rhodamine is used like fluorochrome, we will see red color of lightning. It depends. Anyway, we will see exactly the shape of presumptive causative agent. In case of anthrax, we will see lightning rods in chain. This immunofluorescence test is used like rapid, so it is used for identification of bacteria because of it is specific, because of using of specific serum where antibodies are. It is used for rapid diagnostics of cholera, of whooping cough, of plague, of tularemia, of influenza, for example. So, and it is by using of this test, it is possible to identify bacteria because specific antibodies are used. And a precipitation test named Ascoli test is used for detection of antigens in specimen of animal's body or human body, and maybe just skin of animals. Of course, there are no live bacteria, but there are antigens of bacteria. If diagnosis was not proved during the life of animal or human beings, and it is possible to detect antigens by using of this test, precipitation test. And this test is used for detection of antigens of Bacillus anthracis on some plants where some goods are prepared of skins of animals. And every skin of animal should be checked. Maybe this animal died of anthrax, and to avoid contamination of personnel, of staff, people who works with this skin, this test is done constantly for checking maybe by this test, it is supposed to detect antigens or Bacillus anthracis. If they are detected, the skin should be destroyed completely. We have to place into the flame or maybe into the acid to avoid contamination of people. Realize spores are sometimes due to inhalation of spores, people are infected, and pulmonary anthrax is very, very severe clinical form, not possible to save the life of patients. Biological method is used, and in this case, animals are used like mice, rabbits, and guinea pigs. The specimen is in fact injected into the animals to prove that this isolated pure culture really is able to cause anthrax. And then Bacillus anthracis are able to cause allergic reactions. As usual, bacteria develop hyper-delayed sensitivity. And for detection of it is cell-mediated reaction, sensibilized T lymphocytes are developed when this substance enters first time human body. If the substance is recognized like allergen, and this is kind of immune response, specialized T lymphocytes, sensibilized T lymphocytes, memory cells are developed. And if this substance enters human body again, it is possible to see allergic reaction. And for this, we know some microbes are able to cause delayed hypersensitivity, mainly fungi, some protozoa like Leishmania toxoplasma, and some bacteria like causative agent of anthrax, like causative agent of tuberculosis, leprosy, brucellosis, and tularemia. And commercial preparations are produced. They are named allergens, and they are named after infection name. So in case of anthrax, anthraxine is used for injection, and anthraxine should be injected into the skin of the patient, and we have to wait 3 days to see result. I'm repeating, it is cell-mediated reaction, and it takes time for T lymphocyte to develop inflammation, and if we will see redness and swollenness, means positive result. Treatment: antibiotic therapy, high dose infusion of penicillin. They are sensitive to penicillin. Immunoprophylaxis, live vaccine is used, and veterinary and sanitary measures, isolation of sick and suspicious animals, and we should destroy animals completely, place in the flame or maybe in the acid. Have you any questions? Have you any questions about causative agents of anthrax or tetanus?
No, thank you.
So it is the last lecture this term. It is the last lecture this term, and then you will have 8 lectures coming term, autumn term. Okay. So no lectures, no more lectures. So if you have no questions, I will finish the lecture.